I am Jenita Thinakaran, a PhD student from the Department of Horticultural Sciences, Texas A&M University, College Station. I'm presently working with Dr. Don Henney at Texas AgriLife Research at Weslaco. My research primarily focuses on potato psyllid host preferences, the behavioral and settling ovipositional response of the potato psyllid Bacteria cockerelli, and the transmission of the bacterial pathogen Candidatus Libribacter solanacearum to several cultivated and wild hosts that are prevalent in the lower Rio Grande Valley of Texas. To begin with, I evaluated these cultivated hosts: potato, tomato, pepper, eggplant, and also a weed host, that is silverleaf nightshade, that's commonly present around the valley bordering the potato fields. I evaluated these hosts to find which host would this potato psyllid prefer, with an idea of if it would have better or more preferred other hosts than the main crop potato. So we could eventually use it as a trap crop. In the process, I found we conducted both laboratory and field studies. In the laboratory, we found that the eggplants were more preferred than potato, peppers. However, in the field, we found that potatoes were consistently preferred as long as the host was available. During the end of the potato growing season, however, they switched preference to other hosts such as tomato, eggplants, and the peppers. We are also in the process of evaluating different varieties of potato, how they would prefer one variety over the other. I'm measuring settling in terms of both feeding and oviposition. We found that there is a correlation between the number of adults settling and the number of eggs that are being laid on these hosts. Apparently, eggplant was more preferred in the lab, both for settling and oviposition. Coming to the other aspect of my research also includes evaluating diseased plants, how they play a role in the epidemiology of this disease. I evaluated several LSO infected and the non-infected hosts for preference for these psyllids. And in the lab, we found that the potato plants, when they were infected at 3 to 4 weeks old, they died and collapsed within 3 weeks after infection. And the psyllids did not exhibit a specific preference for the infected or the uninfected host. They settled uniformly on both these hosts. We also inoculated the pathogen into eggplants, peppers, and tomatoes. We found that the tomatoes and the peppers were not very symptomatic until about 6 weeks after infection. However, later on, they became symptomatic, but the psyllids had a choice only beyond 6 weeks. They preferred the infected host on tomatoes beyond 6 weeks. We conducted certain field studies also infecting potatoes and testing the preference of field available psyllids that were available in the open fields, and they did not make a choice for either infected or uninfected. They settled on both these hosts uniformly. There wasn't a specific preference. My third aspect also involves testing this weed host, the silverleaf nightshade, how this could become a potential reservoir host for the pathogen, the Libribacter. How this pathogen could retain itself within this plant system throughout the summer. Is it possible or no? We found that the psyllids are able to acquire the pathogen from the infected silverleaf nightshade within 2 weeks of feeding on the silverleaf nightshade. Presently, I'm testing the roots and stolons of the silverleaf nightshade if they will still retain the pathogen over the summer. A future direction to this course of study would also include evaluating other perennial solanaceous hosts that's available in the southern part of the US, including the potato 3 Solanum erianthum. That is also supposedly a reported host for the potato psyllid and the pathogen.